So I welcome everyone who is already here to the Simcyp Scientific webinar series. It will be an hour that is actually planned for, with a small introduction by me for Melody, and then Melody will give a presentation, and afterwards we can have question and answers. And I'm quite sure you will have some questions for her, and she is willing to answer them. And otherwise, if we are getting stuck anywhere, we also have Josh here to actually help out if needed, right? So we really have a nice scientific communication hopefully going. Initially, all attendees are muted. It might be that you're able to unmute yourself. Otherwise, you can raise your hand, and you can also put comments or questions in the chat, and we can go through that as we go on. The recording today is recorded. Well, the webinar today is recorded. Melody and Josh agreed to do that, and I'm very happy about that. And with that, I'm asking actually Melody to move to the next slide, please, because she's driving the slides. Exactly. And so just to introduce her, so Melody Jin, she is actually fifth-year PhD candidate in Josh Undekant's laboratory at the University in Washington, in lovely Seattle, very nice town. She received her bachelor degree in pharmaceutics from the China Pharmaceutical University, and her background is actually in the experience in the DMPK, including particularly drug transporters and IVIVE. That's where we will hear something about today. So, and let's admit some more people. She has, or is in the middle, pretty much at the end, her PhD thesis for optimizing and validating of the proteomic-informed related expression factor, so the relative expression factor, the REF. This approach to predict the drug clearance, tissue concentration, and drug-drug interactions. And as I already pointed out, Melody is pretty much at the end of her PhD. So if there is anyone here that actually looks for someone like her, she will be available in a few months for a nice job. So you have new raw material here that you can use, and her email address is there as well at the university where you can reach her. With having all said that, she will give us a presentation. And the title of her presentation is Prediction and Validation of Human Hepatobiliary Clearance and Hepatic Concentrations of Transported Drugs, specifically the pitavastatin and glyburide, and she will use the REF approach and the PET imaging approaches. And with that, I'm actually looking forward to the presentation, and we have now quite a nice, decent number of people attending. So far, over 50 people have dialed in, and I'm curious to hear from you. What do you want to tell us about pitavastatin and glyburide and their hepatic transport? All yours.
Thank you, Sibel, for your nice introduction. Hello, everyone. Thank you for attending my webinar today. So you may notice that the title here is a little bit different from the introduction from Sibel. That's because this is a little bit broader title that I will include not only about the REF prediction of the glyburide and the pitavastatin hepatic concentrations, and also I want to cover a little bit about PMUE, protein-mediated uptake effect, which is actually belongs to this optimization part. And they all are the 3 years of project I've been doing under the grant of Simcyp. So here is the outline of today's presentation. I will start with pointing to the importance of knowing the tissue drug concentrations and the drug clearances, and discuss about the current approaches to predict the tissue drug concentrations and drug clearances, and then also pointed to the challenges of the current approaches. And given the current challenges, I will bring up a promising approach, which is called proteomics-informed relative expression factor approach. And then I'll give you 2 case studies from our group, and then ask a question: can the protein-mediated uptake effect, PMUE, bridge the IVIVE discrepancy we have observed in the transport drug clearances? And then I will share with you my goal for this presentation, which is to predict human hepatobiliary clearances and human hepatic concentrations of glyburide and pitavastatin using the REF approach, and then share with you my results, and which is validated by the PET imaging studies. And in the end, I want to expand my scope a little bit to talk about how can we utilize the REF predicted transporter kinetics information in glyburide full body PBPK modeling, and then conclusions and future directions. So I want to start with a question: why it is important to measure or predict tissue drug concentrations. So here, the figure here on the left-hand side shows you the reasons for the phase 2 and phase 3 failures from 2013 to 2015. So from the figure, you can see that the major reasons for the failure in drug development is due to the lack of efficacy and the safety. And this may be likely due to the not achieving the desired tissue drug concentrations or a lack of thorough understanding of the drug concentration versus the drug effect. Therefore, measuring or predicting tissue drug concentrations is very important for us to optimize the drug dosing regimen and the drug efficacy versus the toxicity ratio. So here I want to give you two examples. First example is statin drugs. So as we all know, statin drug they exert their lipid lowering effect in liver. Therefore, knowing the hepatic concentrations of statins will help us to better understand its efficacy. The second example is CNS drugs. So we all know that the restriction of blood-brain barrier really limits our understanding of the brain tissue drug concentrations, which further cause a barrier for us to develop the CNS drugs for humans. So given this, we really want to ask a question: there any routine method that we can use to measure the tissue drug concentration, since understanding the tissue drug concentrations is so important? So in a traditional clinical PK study, only the systemic drug concentrations can be measured, and people tend to use the systemic drug concentrations from clinical study as a surrogate for the tissue drug concentrations. And this can work when the drug is a high permeable drug and only penetrates the cell membrane through passive diffusion. However, if our target drug compound is a membrane transporter substrate, in this case, the presence of the membrane transporters may cause a barrier in the cell membrane. And in this case, the systemic drug concentrations may not equal to the tissue drug concentrations, even at steady state. Given this, we do need a more direct approach to directly measure the tissue drug concentrations. Imaging, for example, positron emission tomography imaging, PET imaging, is a non-invasive, quantitative, and also highly sensitive approach that we can use to directly measure the tissue drug concentration. However, because imaging is so resource intensive and it requires the use of radioactivity, it cannot be widely applied to all the cases. Given this difficulty, as a PK scientist, pharmacokinetic scientist, it's really important for us to predict the tissue drug concentrations using the in vitro or in silico method. Before we predict tissue drug concentrations, it's important for us to first understand the tissue drug concentrations is actually determined by all the clearance pathways that mediate the entry and the exit of the drug to and from the tissues. In other words, to predict the tissue drug concentrations, it's necessary to accurately predict all the clearances pathways. Let's take liver, for example, which is a very important organ for drug distribution, drug elimination, and also drug therapeutic target. According to the free drug hypothesis, only the inbound drug in the blood can enter the hepatocytes, enter the liver, either through the passive diffusion or the sinusoidal uptake transporter. So once the drug enters the liver, it can go back to the blood circulation through the passive diffusion or sinusoidal efflux transporter. or the drug can undergo the biotransformation, form the metabolite, and then being excreted to the bile. Or the parent drug can be directly secreted to the bile through the canalicular efflux transport. So here we can see, if we want to predict the hepatic drug concentrations well, it's very necessary to accurately predict all these clearances that I mentioned before. Then here you can see that our focus kind of switch from predicting tissue drug concentrations to predicting the clearances. So how do we predict clearances? Still, let's take hepatobiliary clearances, which is my focus as an example. Primary cells and physiological scaling factors are widely used to predict the clearances. For example, sandwich culture human hepatocytes is a novel tool which allows the formation of biocanaliculi. Therefore, people tend to use sandwich culture human hepatocytes for the estimation of both uptake and the biliary efflux of the drug. Then how does this sandwich culture human hepatocyte perform? The table here shows the table here is coming from a study where people evaluate where people predict the uptake and also the efflux clearance for different OATP substrates. So here you can see for the prediction of sinusoidal uptake clearance, the in vitro predicted values are always lower than the in vivo observed values, which means there tend to be a systemic under prediction of sinusoidal uptake clearance for OATP substrates. And then let's take a look at the biliary efflux clearance. In contrast, the in vitro predicted biliary efflux clearance tends to be higher than the in vivo observed biliary efflux clearances, which means there's a systemic over-prediction of the biliary efflux clearances. What's more, if we take a further look at this discrepancy between the in vitro and in vivo for both uptake and efflux clearances, you may notice that the magnitude of the under-prediction or the over-prediction of the transporter-mediated clearances for these OATP compounds are different for each compound, which means it's hard for us to find a global scaling factors for our different OATP substrates to bridge the discrepancy. Given this, our group has proposed a promising approach, which is to use the transporter expressing cells or transporter expressing vesicles and the relative expression factor approach to predict the human hepatobiliary clearances. So here you can see we have two important components. The first one is transporter expressing cells and vesicles. Obviously, compared to primary cells, transporter expressing cells or vesicles are readily available and also cheaper. In addition, using transporter expressing cells or vesicles, it will allow us to identify all the transporters that involved into the transport of the drugs, which is important for us to predict the future drug-drug interactions and also the impact of PGX. The second important component is relative expression factor. Of course, because the in vitro system we use is transporter expressing cells or vesicles. So the transporters on the cells or vesicles are overexpressed. And if we want to better predict what is happening in vivo, we do need to consider a factor that corresponding to the difference of the transporter abundance in the in vitro system in the cells or vesicles versus in the in vivo, which is human liver tissues. So to briefly walk you through how we use the REF approach and transporter expressing cells and vesicles to do the prediction of human hepatobiliary clearances. So by using the transporter expressing cells and vesicles, we conduct the uptake or efflux experiments to get the intrinsic uptake or efflux clearances. And then we need to calculate this REF factor, which, as I mentioned before, is the transporter abundance ratio between the human liver tissues versus the in vitro platform we use, which is cells and vesicles. And this transporter abundance information in vivo and in vitro can be measured by the quantitative target proteomics. So once we get this in vitro uptake or efflux clearances and also the REF factors for each individual transporter, we can scale this REF factor and then get a REF scaled intrinsic uptake or efflux clearances. And then by applying for the total protein yield or membrane protein yield and also the liver weight, we can get the intrinsic uptake or efflux clearance for the whole liver. And after we did this prediction, you may ask me the question, how can we validate the predictions of the drug hepatobiliary clearances? As I mentioned before, PET imaging is non-invasive measurement of the tissue drug concentration time profile. So here I'm showing you an example, PET imaging example of the rosuvastatin in humans, which is conducted by our group. So from this figure, you can see that PET imaging really gave us a visualization of the real-time concentration change in the organs. And showing here is liver and gallbladder. And in addition, another advantage of PET imaging is because it's conducted within a short time period. So we have confirmed that there's negligible metabolism of the compound. Therefore, PET imaging is really a good tool for us to really keep track on the transport kinetics of the compound. Until here, you may ask me the question, using an REF approach, you get the prediction of the hepatobiliary clearances. However, it seems that PET imaging can only give you tissue drug concentrations. So how can we connect the tissue drug concentrations and also the clearances together? And to solve this problem, I built up a mechanistic compartment modeling where you can see that we consider the central blood compartment, the liver compartment, and also bile duct and gallbladder compartment. And then we use the sinusoidal uptake and sinusoidal efflux and biliary clearances to connect this compartment together. And one advantage I want to bring up here is of our model is first, in our model we corrected for the liver blood contamination. And second, we enable the presence of sinusoidal efflux clearance, which is normally ignored in other people's liver model. And by building up this model, using a top-down approach, we fit this model into the human PET imaging study. And then we can get the estimated in vivo hepatobiliary clearances. And then we can compare this in vivo hepatobiliary clearances estimation with the REF predict human hepatobiliary clearances. And remember that our final goal is to predict human hepatic concentrations. Therefore, by using bottom-up approach, we can plug this REF predicted hepatobiliary clearances into the model and then simulate the human hepatic concentrations, concentration time profile, which is based on the REF predicted human hepatic biliary clearances, and then compare with the observed human hepatic concentration profile, and calculate the human hepatic AUC and Cmax to see how our prediction is. And then you may ask me a question, how does this REF approach perform? And next, I will show you two case studies still from our group. The first case study is using the REF approach to predict the human hepatobiliary clearances and human hepatic concentrations of rosuvastatin. So rosuvastatin is a typical hepatic uptake transporter OATP 1B1, 1B3, 2B1, and NTCP substrate. And also it has a protein binding degree in plasma about 0.12. Unbound fraction in plasma about 0.12, sorry. Using the REF approach, we predict the human hepatobiliary clearances of rosuvastatin. So first I want to show you the prediction of the human hepatic uptake clearance. So from this figure, you can see that we actually use two different approaches transporter expressing cells REF approach and also the human hepatocyte models with physiological scaling factor approach to predict the in vivo human hepatic uptake clearances. So the solid line here shows you the observed human hepatic uptake clearance in humans. The 95% confidence interval of the observed human hepatic uptake clearances. And the dashed line here shows you the twofold range of the observed human hepatic uptake clearance, which is set as a success criteria of the prediction. So you can see that both REF approach, transport, and also the physiological scaling factor approach under-predict the in vivo human hepatic uptake clearance of rosuvastatin. Although REF approach performs better compared to the hepatocyte models and physiological scaling factors. So given this underprediction, we are wondering like why there is underprediction for hepatic uptake clearance, again, for rosuvastatin. And next, I want to show you the prediction of the glyburide efflux clearance of rosuvastatin using the transporter expressing vesicles. So this time for prediction of efflux clearance, you can see that compared to the observed values, using REF approach, we made a very successful prediction. However, using standard culture human hepatocytes, consistent with literature data, it overpredict the human it overpredict the biliary efflux clearance of rosuvastatin. And once we have collected the biliary efflux clearance and the hepatic uptake clearances, we then, as what I mentioned before, we then try to predict the human hepatic concentrations by putting those hepatobiliary clearances based on REF approach into the model. So you can see that compared to the sanguine culture human hepatocytes, using REF approach, we have a good prediction of the human hepatic concentration time profile. Although it actually a little bit under predicts the human hepatic concentrations. And next, we calculate the hepatic AUC and calculate the predicted values versus the observed values. So you can see that compared to sanguine culture human hepatocytes, REF approach, I would say barely versus the observed ratio of the hepatic AUC by REF approach barely reach the twofold range, which means we have a moderate success in predicting the hepatic AUC of rosuvastatin. So this is a case for the rosuvastatin prediction. The second case is where we use the REF approach and transporter expressing cells to predict the steady state M-bound brain to plasma concentration ratio, which I will call it KPUU ratio, KPUU brain ratio, for 3 different PGP substrates with different fraction transported values by PGP transporter, and also in the meantime have the PET imaging of the brain available, which we can use to validate our predictions. So from this figure on the left-hand side, you can see that for all these 3 compounds, PGP substrates, we have a good prediction of their KPUU brain, no matter how much of the compound is transported by PGP. And this means using the REF approach and transport expressing cells, we have a successful prediction of the KPUU brain for the 3 model PGP substrates. And to here, you may ask me a question. So why there is an under prediction of the sinusoidal uptake clearance of rosuvastatin by using an REF approach? And in contrast, using the REF approach, we successfully predict the human brain KPUU or PGP substrates. And another thing I want to share with you is we also successfully predict the human hepatic uptake clearance of metformin, which is not an OATP substrate, rather it's an OCT substrate and also has less protein binding. Given this difference between the rosuvastatin and the other compounds, we really want to ask a question, is this under prediction by REF approach specific to OATP substrates or highly protein bound drugs? Based on these knowledge gaps, I actually have developed the aims of my thesis into two different sections. First section is I want to optimize the REF approach by incorporating the protein-mediated uptake effect, which I will discuss later. And the second aim is I want to continuously validate the REF approach using other OATP substrates that is besides rosuvastatin. For example, glyburide and pitavastatin in my case. So first, I started my thesis research by optimization of the REF approach, which I investigate the protein-mediated uptake effect, which is a very interesting phenomenon. So protein-mediated uptake effect actually talks about the phenomenon, in vitro phenomenon that when we try to include the plasma proteins in our in vitro experiments, we will see a higher measured intrinsic uptake clearances for highly proton-bound drug. And people name it protein-mediated uptake effect. And the goal of my research is to see what's the mechanism of this protein-mediated uptake effect. And can we use this protein-mediated uptake effect to improve the current IV/IVE for the transport drugs? And I have talked about this topic in previous webinars for several times. So today I won't spend a lot of time on it. I can directly share with you my results. However, our results show that the interpretation of protein-mediated uptake effect of statins, which we use as our target compounds, by both plated human hepatocytes and suspended human hepatocytes is all largely confounded by the residual starting protein complex. To explain it more specifically, for plated human hepatocytes, we found that the increase of the uptake of the statins is due to the non-specific binding of the drug protein complex to the cells or labwares that cannot be completely washed away. However, for suspended hepatocyte system, the increase of the drug uptake in the presence of human serum albumin is due to the centrifugation of the drug protein complex to the bottom layer of the microcentrifuge tube. That is due to the increased density of the drug albumin complex compared to the unbound drug. And based on these results about the protein-mediated uptake effect, I later decided to move my thesis to the validation of REF approach using other transport drugs, glyburide and pitavastatin, because I really want to know that is this endo prediction unique to OATP substrates or just unique to rosuvastatin? Because if it's unique to OATP substrates, then why the PMUE, the protein-mediated uptake effect, doesn't work? So I just want to further validate our REF approach before I draw any conclusions on the endo prediction. So next, I'm going to go to the validation part, which is actually the title you may see from the web page. To predict and validate the human hepatobiliary clearances and human hepatic concentrations of model drug, glyburide and pitavastatin using a relative expression factor approach and PET imaging approach. So you may ask me a question, why did you choose glyburide and pitavastatin? The first reason is they are typical substrates of human hepatic uptake and also efflux transporters, like PGP, like OATP, NTCP, PGP, and BCRP. And second, we have seen the reported under prediction of their hepatic uptake clearances using the primary cells and physiological scaling factors. So we really want to see by using REF approach, can we have a better prediction compared to the primary cells? Last but also very important, for these two compounds, we have their PET imaging in humans, PET imaging studies in humans available, which we can utilize to validate our predictions. And before I start my experiments, I just collected the literature information about the transport information of these two compounds and showing you here. So if you see a question mark, that means there might be some controversial or ambiguous data on this transporter, which definitely needs a further validation of our in-house experiments. So to briefly walk you through the workflow before I share with you results. First, we want to utilize the transporter-expressing cells and vesicles to identify the hepatic uptake and efflux transporters for glyburide and pitavastatin, and then determine the in vitro transporter-mediated uptake and efflux clearances for these two compounds. And next, using the REF approach, we want to predict the glyburide and pitavastatin human hepatobiliary clearances. And then by plug this REF predicted human hepatobiliary clearances of these two compounds into the model that I mentioned before, we want to simulate the glyburide and pitavastatin human hepatic concentrations, which are actually our final goal. And then we want to validate our predictions using a human glyburide and pitavastatin PET imaging data. And here I want to address that. We set twofold of the observed data as our success criteria of the prediction. So next, I'm going to show you the results. First, I want to show you the hepatic uptake of glyburide and pitavastatin using the transporter expressing cells. So here, the two figures here shows you the glyburide and pitavastatin uptake by the transporter overexpressing HEC293 cells. So here you can see glyburide is the substrate of OATP1B1, OATP1B3, and OATP2B1 substrate. And pitavastatin is not only the OATP1B1, 1B3, 2B1 substrate, it's also the NTCP substrate. And then after correcting for the REF factors, we calculate the fraction transported by different individual transporters of the hepatic uptake clearances. And here you can see that for glyburide, around 82% of its sinusoidal uptake, hepatic sinusoidal uptake, is mediated by transporter, OATP transporters, and around 20% of its sinusoidal uptake, hepatic sinusoidal uptake, is due to passive diffusion. However, when you look at the pitavastatin, it seems that 98% of its hepatic sinusoidal uptake is driven by the transporter-mediated uptake clearance, and this passive diffusion only accounts for 2% of its hepatic sinusoidal uptake. And this is uptake information. And next, I'm going to show you the hepatic efflux of these two compounds using transporter-expressing vesicles. So first, we found that glyburide was not transported by any sinusoidal efflux transporters, MRP3 or MRP4. And the literature shows that it has negligible biliary excretion. So for the efflux, I will mainly show you the pitavastatin results. So first, we investigate the sinusoidal efflux transporter for pitavastatin. And our results show that pitavastatin is a substrate of sinusoidal efflux transporter MRP3. In addition, we also found that pitavastatin is the substrate of biliary efflux transporter, PGP, and BCRP. And after correcting for the REF factor, we found that PGP accounts for almost 80% of the hepatic biliary efflux of pitavastatin. And before I show you the prediction results, let me briefly walk you through the hepatic transport of glyburide and pitavastatin. First, glyburide. So the glyburide in blood can enter the hepatocyte either through passive diffusion or the hepatic uptake transporter OATP1B1, 1B3, and 2B1. Once the glyburide gets into the liver, it can go back to the blood through the passive diffusion, or it can directly get metabolized by the CYP enzyme. And then for pitavastatin. So pitavastatin can enter the hepatocytes either through the OATP transporter or the NTCP transporter or the passive diffusion. And once it gets into the hepatocyte, it can go back to blood either through passive diffusion or sinusoidal efflux transporter MRP3. Or the pitavastatin itself can directly be excreted to bile through the biliary efflux transporter BCRP or PGP. So next, I may show you the prediction of the hepatobiliary clearances of these two compounds. So first, I'm going to show you the sinusoidal hepatic sinusoidal uptake clearance prediction of these two compounds. So obviously, you can see that compared to the observed sinusoidal uptake clearances for these two compounds, the prediction for both compounds using the REF approach will predict the human hepatic sinusoidal uptake clearance of these two compounds because the predicted values, they all fall within the twofold success criteria we set before. And next, I'm going to show you the prediction of sinusoidal efflux clearance. So here I want to address that. Remember that for glyburide, it has been shown that it's not a it's not a substrate of any sinusoidal efflux transporter. So we assume that its sinusoidal efflux is all due to passive diffusion. Well, for pitavastatin, some portion of its sinusoidal efflux clearance is due to the MRP3 mediated sinusoidal efflux. So you can see that for both glyburide and pitavastatin, using the REF approach, we can well predict their sinusoidal efflux clearances, no matter it's all due to passive diffusion or it's also due to passive diffusion plus transporter-mediated efflux clearance. And next, I'm going to show you the prediction of biliary efflux clearance for pitavastatin. Very obviously, again, using REF approach and transporter expressing vesicles, you can see that we have a very great prediction of the biliary efflux clearance of pitavastatin. And in the end, I want to show you a very interesting figure where you can see that the figure here shows you the prediction of biliary efflux clearance for pitavastatin before by PGP and BCRP before we correct for REF factor and after we correct for REF factor. So before we correct for any REF factor, these values we have seen, it actually directly coming from the apparent value we obtained from our in vitro platform. So it seems that directly from the vesicles, it seems that BCRP is a major transporter for pitavastatin's biliary efflux clearance. However, after we consider about the transporter abundance difference in the in vitro system and also in the in vivo human liver tissues, now it seems that actually PGP is a major transporter, predominant transporter for pitavastatin biliary efflux clearance. And this comparison really address the advantage of REF approach, which means sometimes the in vitro the values we directly obtained from the in vitro systems without correcting for any factors might be deceiving. So it's important to correct for the REF so that we can know which transporter is the most important transporters, which is further important to inform us the potential DDI, drug-drug interactions. So after we have discussed about prediction of human hepatobiliary clearances, remember that our final goal is to predict human hepatic concentrations of these two compounds. So like I mentioned before, by plugging into the REF predicted human hepatobiliary clearances values into this model, we can simulate the human hepatic concentrations of the compound. So first, I'm going to show you the hepatic concentration simulation of glyburide for a representative subject S0721. So here you can see the orange solid line here shows the simulated values, simulated hepatic concentration profile. And I would say it almost overlapped with the observed human hepatic concentrations, which means we have a very great prediction of human hepatic concentrations of all for this human subject. And because of the for this glyburide PET imaging study, we can get the valuable PET imaging raw data for 7 human subjects from our collaborators. So we can actually perform the simulations of the hepatic concentrations for all the 7 subjects and make the comparison with their observed hepatic AUC. So you can see that the geomean of the predicted glyburide hepatic AUC for all the 7 human subjects fall within the twofold range of the observed hepatic AUC for glyburide in the 7 subjects, which means we have a good prediction, successful prediction of the human hepatic exposure of glyburide. And next, for pitavastatin. So here I want to mention that the PET imaging data of the human hepatic concentrations of pitavastatin is digitized from the current published literature. You can see that again, the REF simulated REF simulated human hepatic concentrations fall within the twofold range of the observed hepatic concentration values. And then we further calculate the hepatic AUC and compared with the observed one. And again, our predicted value, AUC value of pitavastatin fall within the twofold range. So here we can conclude that for these 2 model drugs, using REF approach and transport expressing cells and vesicles and also compartment modeling, we can successfully predict their human hepatobiliary clearances and human hepatic concentrations. So before I wrap up and make a conclusion, I want to expand our scope a little bit more. So we want to see that utilizing this valuable REF predicted transporter kinetics, can we apply them into the full body PBPK modeling using the same SIP to see if we can get a better capture of the human plasma concentration of the compound, for example, glyburide. And this study is led by by the postdoc researcher Ankit Bahara in our lab. And the reason why we want to utilize this REF predicted transporter kinetics in the full body PBPK modeling of glyburide is we check the current published glyburide PBPK modeling. In their input parameters for the hepatic transporter, it either directly use the sanguine culture human hepatocyte data in their table, which I have shown before, definitely has some disadvantages. Or they assume that OATP1B1 is the only hepatic uptake transporter, which again is not quite correct based on our in-house data. And because of this erroneous input, to better capture the plasma concentration time profile of glyburide, we may need a scaling factor around 2 to 10 to really achieve the goal. So our goal is to see, by utilizing the REF predicted transporter kinetics that I generated in the PBPK modeling using the same thing, without scaling factors, can we be able to capture the plasma concentration of glyburide? And I'm going to show you the results of the prediction of plasma concentration time profiles of glyburide in the healthy populations. So here from these 3 figures, you can see that for both IV bolus, IV infusion, and also oral administration, using our REF predicted transporter kinetics information into the input parameter for the PBPK modeling, without any scaling factors, we can successfully capture the plasma concentration time profile. And if we take a further look at the predicted values versus observed values, which we call PO ratio of the Cmax and the AUC for glyburide, you can see that the PO ratio for Cmax and AUC fall within the bioequivalence criteria, which means we have a successful prediction of the plasma concentration time profile of glyburide without any scaling factors using our REF approach. So in the end, next, we want to see that using the REF predicted values in the PBPK modelings, can we also be able to capture the drug-drug interaction condition? So we collected the studies where the subjects were co-administered with glyburide and also the rifampin, which is a very typical OATP transporter inhibitor, but CYP enzyme inducer. So we want to see that using our REF input values, can we be able to predict the AUC change with and without co-administration of rifampin for the subjects? So you can see that compare our predicted AUC ratio value with observed AUC ratio value with the co-administration of rifampin. The PO ratio is ranging from 0.87 to 1.35, which again shows that we can well capture the drug-drug interaction condition using our REF predictive values as the input of the PBPK modeling for glyburide. So now I want to make a conclusion of today's presentation. So first, accurate prediction of tissue drug concentrations is important to better understand the PK/PD relationship of transport drugs. And we have shown that REF approach, which is informed by quantitative target proteomics, performed relatively well in predicting human hepatobiliary clearances and human hepatic concentrations of glyburide and pitavastatin. And based on this success, and together with our previous success, the REF approach can be used in the future to predict transporter-based drug clearances and tissue drug concentrations to inform the dose selection in the clinical trials. And what's more, our previous investigation on PMUE, protein-mediated uptake effect, shows that the observed protein-mediated uptake effect using the plated and suspended human hepatocytes is largely confounded by the residual study albumin complex. Therefore, mechanism that other than protein-mediated uptake effect should be explored to bridge the modest IVIVE gap for OATP drugs like rosuvastatin. In the end, the REF predicts the hepatobiliary clearances of glyburide can be applied to the full-body PBPK model to successfully capture the plasma concentration time profiles of glyburide with and without co-administration of rifampin. And here I want to address that our eventual final goal is not only just to predict the DDI or just to predict the plasma concentration in healthy subjects. There are other lab members in our lab. They are also investigating the drug plasma concentration time profile and their time profile change and their inflammation or in the pregnancy woman, where we can definitely utilize the current knowledge about transporter abundance and transporter activity, and also even enzyme abundance and enzyme activity we can apply this knowledge into the PBPK modelings and to see if we can also being able to predict the drug plasma concentration time profile for these special cases. And with that, I want to shout out to many people. First, my PhD supervisor, Dr. Josh Wunakat, for his support and help and guidance through all my PhD work. And also, I want to thank my PhD thesis committee, Dr. Nina Esser-Heron, Dr. Ken Samo, Dr. Shin Mao, and Dr. Joanne Wang. And also, I want to thank my collaborators, Dr. Ankit Bahara, for his help and guidance with the PBPK modeling and also some of the vesicle studies. And also, I want to thank Dr. Saloni and Dr. Nicholas for their generous support of the PET imaging study of glyburide in humans. And also, I want to thank Dr. Flavia and Dr. Simon for their help with the PET imaging data analysis. And I also want to thank Solvo Biotechnology for their generous support of transport expressing cells and vesicles. And also, I want to thank Gilead Science DMPK department, Dr. Yurong Lai, Dr. Kazuya Ishida, Dr. Xiaomin Liang, for their help and guidance for the PMUE work on suspended hepatocytes. And also, of course, I want to thank my funding, especially Certara SimSip grant for the generous support for my 3 years of research. I have really learned and gained a lot through the project. And also, I want to thank UW RAPT, which is supported by Gilead Science, Amgen, Johnson & Johnson, and Takeda. And thank the Department Award, JP Wang Award, Danny Shen Award. And in the end, I want to thank Department of Pharmaceutics, University of Washington. And with that, I'm happy to take any questions from the audience.
Thank you very much, Melody.
Thank you.
You left us 10 minutes time for questions. That's highly appreciated. And you already see people are exploding. So
Oh, I can't
It
see
was
that.
enjoyed. We actually
yeah.
do have questions. So
Sure.
Gaurang has raised his hand, and I think go ahead and have your question. Can you unmute yourself?
Yeah, it was by mistake.
Yes.
Yeah, sorry.
Oh, that was a mistake. Okay, then I saw Shinji, you had your hand raised.
Hello, can you hear me?
Yes, we can.
Yes, I can hear you.
Yeah, I'm interested in like I believe pitavastatin like PGP and BCRP balance like from in vitro to in vivo. Then do you have any plan to demonstrate this difference in vivo?
Sorry, would you mind repeating your question again?
Yeah,
The
so
difference?
eventually like kind of starting BCRP substrate, right? These are typically we
Yes.
believe, but if you apply kind of left approach, then you can show like PGP is more important than BCRP.
Yes.
Yeah. And then do you have, so this is still a prediction. So do you have any plan to demonstrate like kind of like this difference in vivo?
So you mean, do I have any like in vivo validation to show that it is like predominantly transport? Oh, that's a great question. I haven't thought about it. I'll take a look at it. Currently, I don't have information about that, about the in vivo information. That's a great question. Thank you. But sorry.
Thank you.
Yeah.
But Shinji, I think this is also problematic to get because you're looking at hepatic transporters. If you look at intestinal transporter side, you might get easy on, but on the hepatic side, I guess they would need PET imaging in order to have the liver concentrations. We are back to that circle, right? So
we also,
so this is a reason why I asked how we can demonstrate. It's very difficult, I thought.
Yes.
So I'm curious
really
about
needs
the inside.
It may need a selective inhibitor for PGP or BCRP so that we can know the information. I remember I used to take a look at the in vivo DDI study. I don't have the memory about this PGP or BCRP information from any DDI studies. So yeah, I don't have the answer for your question. But yeah, thank you for your question.
Thank you.
I agree with all those comments that Sybil and Melody made. This is Josh Nadkat. I think you'd really need PET imaging and selective inhibitors of PGP and BCRP. That would not inhibit the uptake transporters or any of the sinusoidal efflux transporters. So, you're getting into like selectivity that's really, really tough to get.
Yeah, and we don't have any knockout, right? In the dog, we would have knockout MDR1, right? They wouldn't have it. So theoretically, in the dog liver that has mutant on the MDR1, then you would have only BCRP, and there you could look at differences. Without that, you need to have an inhibitor or that is then selective. But otherwise, I wouldn't know how you can get there. Shinji, it's a very good question. Maybe I can refer actually to the paper that from the ITC consortium was written last year, where they looked in general if biliary inhibition of PGP and BCRP is actually relevant in clinical studies, and we couldn't really find anything decent for that. So I think that is actually something open, Melody, where you can still have some research on, as we have discussed. Okay, are there any other questions from the audience? Anyone would like to have something more? Well, it's at the moment very quiet. Armin, please go for it before I put my questions. Armin Sepp. Go.
Oh, thank you very much. I'm relatively unexperienced in this field. So, but you mentioned protein-mediated enhancement of protein-mediated uptake. Can you briefly recap how does it work? And in the experiment, you mentioned that there was a residual binding to albumin, and that was presumably after the medium was washed away, where the medium, the albumin concentration roughly is 10% fetal calf serum. So albumin concentration would be 50 micromolar. After washing, it would be probably nanomolar range. how quick is the equilibrium one would expect any complex to fall apart within seconds, if not minutes?
Yeah, I'm not sure if I totally understand what you mean. So you want me to walk you through again about the protein mediated uptake effect finding we have?
Just one,
Yeah.
one sentence, or 2 sentences.
Yeah, let's take a plated cells as an example. So we find that the increase of the drug uptake, the so-called increase of drug uptake in the presence of plasma proteins like human serum albumin is
Right.
due to the non-specific binding of the drug albumin complex. The binding to the cells or labware that cannot be washed away, so that they kind of stay left over in the plate. And when we lyse the cells, we're taking those non-specific binding into consideration of the drug uptake. But they're actually not the real drug uptake. They are actually just non-specific binding.
Artifact.
Yeah.
Right. Thank you.
Basically, you get the free fraction at the end wrong because compound is bound to equipment and you're not really correcting for it correctly.
Yeah. Actually,
Okay.
I have a sure. No
Yeah.
problem. Go ahead. Yeah, I just
No,
want
go.
to add. Yeah, I just want to add up one point. I have a good animation here shows you like how this non-specific binding can occur and being taken into consideration of the drug uptake. So you can take a look at this figure here.
Great. Thank you.
Yeah.
Okay.
Yeah.
So I don't see any other hand raised. I would have one question.
Sure.
Okay. We assume everything is perfect by understanding abundances, right? Abundances in vitro versus abundances in vivo, right? I mean, this is not really a new concept as that, but it failed predominantly because people had not very good measurements, right? You need to have good measurements, otherwise you're doing obviously wrong corrections.
Yeah.
And you had a compound, pitavastatin and OATP1B1. Now, in between of populations, you do see really different concentration profiles. So, and I assume that your profiles that you picked up were for Caucasians.
Pitavastatin is from Japanese.
Those ones for
Males.
Japanese.
Yeah, we
But
digitized from a Japanese
there
study.
are
Yeah.
differences, concentration time profile differences between Caucasians and Japanese.
Yeah.
And they come from the OATP1B1. At least that is so far the discussion, right? So you get significant difference, but you don't really see differences in the abundance. So how do you explain that? What would be there for the additional difference between those profiles?
Yeah, that's a great question. Actually, I think about the difference between different populations. So first, I want to clarify that the PET imaging for pitavastatin is we can only be able to get the data from the Japanese populations. However, the REF values, especially the in vivo human liver results, I assume is mostly coming from the Caucasian populations. So yes, there might be some discrepancy between the in vivo observed population and also the REF values, the population that we use to calculate REF values. And I try to think about, try to correct for this by searching literature, like is there any, for example, proteomics results of the transporter abundance difference between different populations. However, I was not able to get this abundance. So in my study, I assume that the transporter abundance, for example, the OATP and the PGP and BCRP using in my study, the transporter abundance between Japanese and Caucasian population are the same. That's one of my assumptions because I couldn't find the literature data to support the difference you say on the transporter abundance between populations.
Yeah, that's exactly. I don't think that there is a difference in abundance. There is a difference in activity. So there is something different because the abundance differences I haven't seen so far. And for the few samples that we have from the proteomics, we don't see this difference. So, but there is obviously an activity difference. So that's why I was asking for. And I think that is still something that we need to go further on.
Yeah,
I mean,
I agree with
Yuichi had
you.
obviously his publication on it to show you have a correction factor of about 0.5. Which you can use in order to align Caucasian and Asian profiles for OATP1B1 compounds. But the underlying mechanism of that is still something that is open. I actually saw there a hand risen and then it was gone. So who was that? Still last question. You have the chance to come back.
So may I say something here, Sybil?
Yes.
To follow up your point, it's a really good point, and we've thought about this as well. There is also the issue of genetic polymorphism that varies across the populations, you know, and there is, of course, as you say, data in the literature that says that the statin PKs in the Asian population, you know, is different than in the Caucasian population, and some of it might be explained by the differences in genetic polymorphisms, you know. So, of course, we could include some of the genetic polymorphism incidences, so to speak, you know, the frequencies in our data to look to see whether that can, you know, what the difference would be in terms of the predictions, you know. But of course, we are under predicting, so we're not over predicting, you know, so that's the other thing that we have to keep in mind in terms of the uptake clearances, you know, so it doesn't quite go in the direction of the genetic polymorphism thoroughly contributing in that way. You know?
Yeah, but there are data actually enough from the genetic polymorphism to separate this out. This is what Yuichi had used at that time. And there were data for the pitavastatin, that's why I asked for it. So it might be worthwhile to see how the profile, because you use the SimSub simulator there, so you have the concentration time profile, and therefore you can also have the liver output, the liver concentration, and you can see how that are actually matching in between of different population. And with the different phenotypes and frequencies, they're already in the simulator. So you can see how they match actually with your observation. So that might be something to look at. Okay, now we are 2 minutes over, but Amaka raised her hand. So maybe you have then the last question because I asked for it, if you don't mind.
Thanks, Abila. Thanks, Melody. So my question is around the REF, the REF approach. And probably because I missed it, but so in the REF approach that you used, do you have a global REF for the uptake and the efflux separately, or did you have independent REFs for each transporter?
Yes.
And then a follow-on question then is, relating your REF to the literature review that you did, where you saw that the scalings that are applied for the uptakes are usually greater. Did you see that same trend from your data? Because you didn't give us your REF values, or maybe if you did, I probably missed it. But I was just trying to see how do your REF values that you estimated, how do they kind of like tally with the REFs from your literature data, your literature review? Sorry.
Yeah, so that's a great question. So for REF value, the REF value, we have it for each individual transporter. Or to say it more specifically, we have the REF value for each independent experiment, because we consider about maybe, you know, even for the same cells, we may have the transporter abundance fluctuation across different passages. So we actually measure the transporter abundance for each of our experiment to calculate the REF value for each transporter. Yeah. And for the comparison between our value with the literature value, I don't think, so first, I don't think there is much REF value or information from the literature. And second is, I think there's no much like meaning of doing that, because the REF value really accounts for the difference, the ratio of the transporter abundance in vitro and in vivo. So if another lab say they have their own in vitro systems. Of course, they may have a different ref because their in vitro transport abundance may be different from ours. So, yeah.
I think you really made the point there. And it is important to have the individual ref values for your laboratory and your experiment.
Yes.
If you want to have a list of them, they're in our help file in the simulator. I'll put them in the end. And we also published last year on a list of those ones that are available. And exactly the point. You need to have reproducible assays, and you actually need to measure it for each of those ones. And until you are actually so experienced in your assay where you know where your bandwidth is.
Yes.
But that's pretty much not there. So I think therefore, and also over the time, I guess in a few years when these experiments have been more established in each laboratory to see actually how this value is changing over time and within a laboratory, that would be an interesting thing then to see how reproducible an assay is, and then you can establish it in another laboratory.
Yes.
Yeah.
Thank
Okay.
you.
I think with that one, I will close off here. Melody, I thank you very much for giving this presentation.
Thank you.
Thank you very much for doing all this work also with the GPS, right? So, and with that, I probably just want to say that we have this SIMSIP grant and partnership scheme since a few years now. You were one of them that went through, and we want to have this
I feel
now
grateful.
as the end of their PhD. So when they have everything put together, that it's also becoming public as a whole tool, because we would really like to have this research as a public, you know, everyone should benefit from this. You get hopefully your PhD soon and then a nice job. And we got all this lovely research from you. So I already would like to thank you for all your work you have done with it. And we hope that in the next round of PhD or postdocs that apply for this grant, we get as good candidates that also do future research. Thank you very much.
Thank you. Thank you, Sivan. Thank you, everyone, for attending the webinar and asking me the questions.
Yes. Thank you, everyone on call and having all these great questions as well. Okay. With that, I will close this WebEx. And if you have any questions, you know where to reach us. Thank you. Bye.
Bye-bye.